Hello, everyone, and welcome to phyloseminar.org. The current theme is on machine learning approaches to phylogenetics, and this is the first talk in a series of 3 talks on that topic. Please use the YouTube live comment box to ask questions. Today's speaker is Anton Suvorov. Anton received his undergraduate training in entomology from Moscow State University, Russia. He then did his PhD at Brigham Young University, Provo, at Seth Bybee's lab. Currently, he's a postdoctoral research associate at UNC Chapel Hill in Dan Schreider's lab. Welcome, Anton, and thank you for participating.
Yeah, thank you, Eric, for a nice introduction, and welcome to the seminar. So today I will be talking about applications of machine learning to tree inference. So, but before we start, so I'm going to talk about this. I'm going to tell you what my talk is going to look like. So it's going to be split in 2 parts. So the first part, I'm going to give you a brief introduction to machine learning and deep learning in general and its applications to evolutionary biology. And the second part of the talk will be dedicated to estimation of tree topologies from multiple sequence alignments using deep learning. So let's start from hierarchy of machine learning. So here we have a domain of artificial intelligence. Then we have machine learning, and we have this like a modern deep learning set of algorithms. So here's the hierarchy of machine learning itself. So there are different methods. So the most popular ones, it's this unsupervised learning type techniques. So basically, it's when we want to, like, for example, cluster some of your data. So we don't know any. We basically want to find some hidden structure in your data. And from human perspective, I can give an example. So I want to learn a new language, and the only thing that I have is just like I can only listen to the radio. So and for example, I'm trying to learn Chinese, and I'm listening to the radio. So at the beginning, I don't know anything, but after listening over and over again, I can find some patterns and some structure, right? So the supervised learning domain, it's basically when we have our input data and we have certain labels with that coming with that data. So we have like X and Y, right? So, and we have certain algorithms that we can train those supervised learning architectures or algorithms to predict Y. Given the X, we can predict Y, right? And we have reinforcement learning, and I'm not going to talk about this. But those approaches are very interesting and curious. So supervised approaches can be further divided into classification problems or regression problems. So a classification problem, it's basically when we want to classify certain things like cats, animals, pictures, or want to, for example, if we're looking at like cancer cells under the microscope, we want to say whether your cell is cancerous or not. We have basically just like discrete labels for your data, right? So for regression problems and tasks, we basically were trying to predict continuous value for Y. So we have certain X observations, and then we want to predict Y, which is a continuous value. So, and this is a little recap. Let's think about linear regression for a moment. So this is the problem. So we have our features, X's, and we have our labels, Y labels, right? And this is our observations. And eventually I want to estimate certain parameters in our model in order to predict Y for this particular case, right? So what we do, we have our linear model, we have our weights, and we can estimate those weights, right? And we have our error term, which can be distributed according to normal distribution with unknown variance. So in order to estimate those weights from this model, we can just simply use maximum likelihood approach. And in fact, for this simple example, we have analytical solutions. We can derive the estimators in a closed forms. We just basically we do partial differential. We just differentiate log likelihood function with respect to every parameter, and we have analytical formulas for every parameter of interest. And then once we estimate our parameters from this features and labels. We can, so those are blue. In blue, we have those estimated parameters, and then we can basically plug these values with a known y, and we can estimate y, right? And now we have another type of regression problems. Those are, for example, logistic regression that we can use for classification. So again, this is the problem. So we have our features, x1, x2, for example, and we have labels. We have labels for our data set, and labels are discrete, like 1 and 0. For example, I don't know, like cancerous and non-cancerous cells. And here we want to predict the label for these two examples. So this is how we set up the problem, the model. So we have our linear equation, and we have, again, error term that is distributed according to logistic distribution, like standard logistic distribution in this case. And if this, the value of this equation basically bigger than 0, then we can assign label 1. And if it's 0, if it's something else, so we can assign label 0. Unfortunately, we can't derive the estimators in a closed form for this problem, but we can use some optimization methods like Newton-Raphson technique and estimate our parameters. But now let's look at the same problem under slightly different angle. Let's say we have a biological neuron, right? And we have certain inputs, x1, x2, x3, x4. And those inputs are coming through dendrites, and our neuron is processing this data somehow, and then it produces some output. And our output is going to be very simple binary output. It's going to be this neuron either fires or it doesn't fire at all. So how we can model this mathematically? So in 1943, McCulloch and Pitts, they came up with this very, very trivial and simple model of the neuron. So we have our inputs, x1, x2, x3, and xn, like as many as you want inputs. Those inputs, according to their model, they can only assume values 0 or 1. Then we have so-called input function. That's kind of like our processor. It's basically how kind of like our neuron process this input, right? So it just sums up those inputs, right? And then we have our activation function. So activate, and it's hard. It's like activation function with hard threshold. So basically, we have certain input here, some value, and then we should determine this threshold, like for example 5 in this case. And for example, if our output of input function is greater than 5, then we can assume then our neuron will fire, which is equal to 1, or it doesn't fire, which is equal to 0 if that value is smaller or equal to 5. Unfortunately, this present form, this model can't learn anything. It's just like we can just feed our inputs and get our output, binary output. So this is when revolution started with this Perceptron model that was proposed by Rosenblatt in 1958. So, and this is the model itself. As you can see, it's very similar to our previous neuron model. So we have our input, we have our inputs. For simplicity, I have 3 inputs here. We have input function and we have activation function. What's revolutionary here is that now we have parameters in this model. The parameters in this model are going to be weights that associated with each input here. Another very significant difference is that here we have so-called bias node. And I'm going to tell you why we need this bias node. Basically, it's kind of like it's the inputs of 1 multiplied by the weight 0. So, and again, we have activation function. So within his Rosenblatt definition of perceptron, he used sine function. So sine functions, basically, it's defined here. It's very easy. You just plug your output of your input function here, and it's going to be either -1, 0 or 1. But for simplicity, let's assume that our sine function only speeds -1 and 1. We don't have cases when we have 0. When the input function output will equal to 0, we don't have those cases, right? So in the output, again, it's binary. It's -1 or 1. So, and let's set up the problem again. So we have our training data set. We call it training data set. This is what we observe, for example, and we want to distinguish between cancer cells and normal cells. And so cancer cell will be associated with label -1, and normal cell will be associated with label 1. And for example, X1 and X2, some properties of those cells, like cytoplasm volume, for example, that's X1, and some other thing, it's X2, right? And then we have like what we want to do. We want to train our Perceptron model to estimate those parameters, those weight parameters, and then we want to pipe this new. We want to basically predict Y. For these new cases, new examples, right, by passing this X1 and X2 through our trained network. So how we can do that? So we have to come up with certain learning rule, and it's called Perceptron rule, and it's very easy in this case. So basically, let's go through every example of this training data set, and then we see when we initiate some weights. And then when we see when the label is not corresponding, the output of this network under certain weights is not corresponding to the actual label in our training data set, we can update our weights according to this simple formula. So we have our old weights, and we plus this little term that is basically, we're just multiplying x's by label y. And this is how we get updated weights. So, and we cycle this, we do this procedure iteratively through all the training data sets, through all the lines, n lines, right? And then we estimate our, again, our weights, and then we want to predict this y. So, and here's what's happening during training procedure. So we have this, let's assume that our examples are linearly separable. We have our normal cells and cancer cells, and we have our, it's called like a decision boundary, right? So, and with every iteration through training data set, Perceptron is learning this decision boundary. So it's like moves this boundary within this space, basically. And eventually we want the boundary that will like perfectly separate normal cells and cancer cells. And going back to this bias node, this was weight 0. Basically, bias node is needed in order to change angle of this decision boundary. That it's not only moving up and down, but it's also like moving left and right, changing by changing its angle. So unfortunately, Perceptron learning rule is not very general, and here we're gonna derive so-called delta rule. It's more general updating rule. And we're going to use the notion of gradient. So here's our problem again. Our data set, our training data set, we have cancer cells, we have normal cells. And notice how I changed the labels now. They're going to be, every label, it's a vector 1 and 0. The first position will correspond to cancer cell. The second position will correspond to normal cell. And when 1 is here, though, it means we have cancer. And if 1 in the second position, then it's normal cell, right? So for simplicity, I have just one input here, x1. We have our Perceptron. But what I'm going to do here, I'm going to use different activation function. Instead of using our threshold sine function, I'm going to use a soft function. In this case, that's going to be a very simple logistic function, standard logistic function. And the output of this neuron, it's not going to be binary output. In this case, it's going to be continuous value. And because this the function runs from 0 to 1 on y-axis, we can interpret this this value as a probability. So, and depending on this value, if it's, so for example, if this value is greater than 0.5, then we can assign that that's going to be our cancer cell. If it's smaller than 0.5, we can assume that's our normal cell. So what we need, second, we need a certain error function or loss function. In this case, I'm going to use very naive and simple mean squared error function. And we want to optimize this error function. We want to make error as close to 0 as possible, right? We want to minimize it. And this, I just write it out for you like here. So this is basically O is our activation function, and then it's just basically, if we use the logistic function, then we can write it out like this, right? So again, using the notion of gradient descent, we can differentiate this error function with respect to our weights, and we basically can derive our delta rule using the gradient like so. And so basically, within the delta rule, again, we have our old weights, and they're going to be updated according to the value of this of the differentiated function, right? Our differentiated error function. And also, it's very important to note that activation function and error function itself, they should be differentiable. That's a requirement. If they're not, so we will have a problem. We can't use them for in a learning framework. Here we have certain, it's called learning rate hyperparameter, that we can tune this parameter in order to adjust our learning. So, and here, imagine this is like our error surface with respect to our weight parameters, weight 0 and weight 1. And with, for example, we pick some learning rate hyperparameters. Suppose it's like, I don't know, 50, like 10 or something. And we can just basically going through every example in our training data set, we can update those weights, and we can hopefully will find the solution that will minimize our error. So this is, we pick this one. And probably the error rate, the learning rate is too high. We can change it, and we can make it more optimal and walk through this space like so. So it's more optimal. So what we want to monitor when we're training our algorithm, this modified perceptron, it's basically want to monitor loss, and in ideal situation, we want loss to go down, right? To go to 0. And so when we're going through all the training data set, so one run through the entire data set, it's called training data set, it's called epic. So here the epic, right? And so we want, again, loss going down, and accuracy of our training procedure should increase. This is what we're aiming for, right? So before I describe the perceptron, and that perceptron can only do binary classification, but can we do multi-class classification? So for example, instead of just having cancer cell, normal cell, we have, like, for example, some intermediate cell that is like changing from normal to like cancer in transition phase. And of course we can. So here you go how we do this. So we have, again, our bias node, our input x1, and instead of our one neuron, we have like kind of like 3 neurons, right? And with corresponding input function. And then we have our widely used, it's called softmax activation function. Basically, it's kind of like you can think of this as like a shared activation function between those that kind of like combines the input of those different the outputs of input functions, and it predicts the vector, probability vector, basically. And so those probabilities are going to sum up to 1, And again, this is a simple example. So for example, after some iteration, we got this like probability 0.7, 0.2, 0.1. And according, for example, we have probability 1 is equals to 0.7. This is the highest probability. And so we can assign label. That's going to be our cancer example. Yeah. We can use more sophisticated and we can use other loss functions, and one of the widely used loss function in classification problems is called categorical cross entropy loss function, as described here, and that's going to be our minimization objective. And again, we can use our learning rule to update our weights when we go through every training data point. And we can update actually those weights. We can update them every time we go through every single example, or we can update them through so-called like a batch algorithm, when we can pick up, like, for example, let's update our weights only after going through 5 examples in this data set. Or we can update all the weights after we go through the entire data set. So let's talk about a little bit about perceptron convergence theorem. So what it says basically, it's after finite number of iterations, perceptrons estimates weights that classify all the examples perfectly. And this theorem can be applied for only for classic Rosenblatt perceptron. And here's the reminder, the structure of our input function and activation function. So basically, this perceptron can only perfectly classify this, for example, these data sets that are like red crosses and blue circles, only if they're linearly separable. And this is our decision boundary or hyperplane, whatever you want to call it. So, and eventually when we learn those weights, we eventually will find this, this line that will perfectly separate those examples, right? But how about this case when we can't linearly separate our data? What are we going to do? Is there any, like, can perceptron learn this? Actually, it can. So let me give you like this very simple example. So we have those blue circles and red crosses, and imagine like we have to come up with certain decision boundary. And as you can see here, so probably circle will be a good decision boundary, right? So and if we remember the equation for circle, so we can actually perform transformation of a nonlinear transformation of our inputs, and after doing this linear transformation, we just take a square of those x's. We can linearly separate this, right? This particular data set, and within machine learning framework, how we can achieve this nonlinearity. So we can achieve this through 2 things. So first, we can use some activation functions that are nonlinear. And if you remember this, our logistic function, it's basically, it kind of like changes our input. This h is going to be input. Remember, it's like a weight multiplied by x's. So this transforms our linear input into very nonlinear, to a non-linear domain. And the second practical approach to increase nonlinearity, it's basically, why not to use more layers in our neural networks? So here's the example of our neural network, and we call it multi-layered perceptron. So basically, we're just putting together different like perceptrons together, and we're kind of like chaining them together, right? So we have our input layer, we have hidden layers, and think of this as like it can be like it's just classic perceptron, and this hidden layer too, and then we have output layer. So within classical framework, we had only input layer and we had only output layer. So, and by adding those hidden layers, we're increasing complexity of our basically we're increasing non-linearity by adding more hidden layers. And again, that's it's a small reminder, the anatomy of each neuron here. We have our input function, we have our activation function, and this is the example of very widely used activation functions, and one of the most popular and very well-behaving function in practice, that the ReLU function, and people use it all the time. The problem is, okay, that's all great, but can we use delta rule or our learning rule to update weights through the networks? Unfortunately, we can't in the present form. But what we can do, we can use so-called backpropagation algorithm. So if we have, for example, 784 inputs our axis, so we can like feed them through the entire network here. And then we can apply, using this backpropagation algorithm, we can update our weights. And we can do it iteratively, right, through this network. And this is just a kind of like demonstration how learning procedure is like achieved and what's going on. So we have our very simple network. We have one hidden layer, one input layer, and one output layer. This is basically, this is a loss. This is loss, and this is going down with all the inner iterations, and this is the red and blue dots are examples, and this blue and red regions correspond to the different hyperplanes. And as you can see, it's like kind of like our algorithm converging. It's learning correct weights to correctly distinguish distinguish between red and blue dots correctly. There are a bunch of different actually machine learning algorithms besides the neural networks. So there's widely used, like for example, linear support vector machines are very popular machine learning methods, or random forest is widely used, or decision trees. And as you can see, they have like different, they approximate our hyperplane slightly different. But let's set up a new problem. So before we were working with those like feature vectors when we had like axes everywhere, right? But how about like doing classification on pictures? And here's an example. We want to classify cats, sunflower, and dog, right? So we want to classify those. We want to train algorithm, our network that will be able to distinguish between cats, sunflower, and dog, for example. And one of the very naive procedures will be like, imagine we have this 3x3 image, and those numerical values correspond to some, for example, pixel, like some abstract pixel value. What we can do, we can linearize this picture, or in machine learning, we use the term flatten. We can flatten that input, right? Like so, 1, 3, 2, 1, 1, 1, 4, 2, 3, flatten it, and we can just pipe it through our neural network. The problem with this approach is that it doesn't work really well. The accuracy of this network is not going to be great. And very recently, I mean, relatively recently, 9 years ago, this team, led by Krzyzewski, they came up with very, very simple procedure that is very effective. And this procedure basically created convolutional neural networks. So the idea is like, it's pretty straightforward. What we have here, so we have so-called convolution layer. So green is our input, our picture, for example, and we have this this feature extractor, as you can see, this orange square, 3x3 square, and it has weights like 1, 0, 1, 0, 1, 0, 1, 0, 1. So, and basically we're striding this kernel or filter or feature extractor, whatever you want to call it. We stride through this image, and we are getting convolved feature by basically, it's just simple by multiplying every cell of a feature by every cell of this image, right? It's just simple Hadamard product of two matrices. This is more defined. We can define it more formally, like so. And the second layer we can add to this, it's called pooling layer. So what pooling layer does, it's so once we got this convolved feature, we can have pooling layer. So we define some matrix, like for example, this pooling layer 3 by 3. And we have, for example, max pooling type pooling layer. So basically what it does is just like when it strides through the convolved feature, it just picks the largest number and it plugs it into our pooled feature, like so. You can ask like what kind of features we're like or parameters we're going to learn here. So it's pretty simple actually. So parameters that we're going to learn, those are convolutional layer parameters, because when we're initiating our kernel, we also initiating random weights first, and those weights are going to be learnable, like, right? So this is like one of the typical architectures, convolutional neural network architecture. For example, want to classify different pictures of numbers from 0 to 9. And then we initiate 10 different filters. Then we have pooling operation. Then we have another convolutional layer, another pooling layer. We repeat those like procedures, right? We kind of have this like deep network. And then at the very end, once we arrive to the pooling layer, we just again flatten the entire output, the entire pooling output, and we pipe it through, we pipe it through our fully connected layer, which is our classic multi-layer perceptron. We just pipe it through here, and we can get an output. In this case, it's going to be classification problem because we're classifying between different pictures from, again, numbers from 0 to 1. And here's the example, like what's happening during convolution and pooling procedures. So this is our original picture. It's 8. And then we perform convolutional layer. And as you can see, we're kind of like transforming our original space into some other abstract spaces. And eventually, like, for example, after pooling layer number 2, we flatten this output, and this is our flattened output. You see there's like flattened pixels. And so supposedly every layer will learn certain abstract representations of our original picture that will be used in learning algorithm and can be learned. This is very popular kind of like problem to distinguish between different faces. And there's a competition, ImageNet competition, and basically the year 2012, as you can see, the implementation of this AlexNet, or the convolutional neural networks that was proposed to Krzyzewski. It boosted accuracy immensely. And then this is where next revolution in machine learning started. This is the time when they invented convolutional neural networks. And then right now, like in 2016, the error for image classification, for faces classification, was nearly 0. Let's briefly kind of like combine everything what we've learned so far. We kind of like I'm going to give you like a general framework of machine learning, like general supervised learning problem. So suppose we have unknown input distribution for our axis, right? And then we have our so-called target function that we want to approximate with our machine learning algorithm. And we can define it. So basically, it's like ideal function. When you plug x's to this function f, you can accurately predict y. And we can also we can add noise, and we can add noise to this function, and we can derive this kind of like we can define it in a probabilistic way. And here, how we're getting our training examples. So we have our x's with corresponding label y. And then we have our hypothesis test, which is basically just a set of different weights. that we're trying to find the best combination of those weights, right? And we have our learning algorithm, our learning procedure, one, like, for example, network. And then we have our error measure, which is our error function or loss function that we can use. And then the goal here is to approximate this function f here, right? So, and the final hypothesis, it's going to be basically the trained network with those weight parameters, right? And we're hoping that it approximates well to our target function. And of course, we have this unknown distribution can supply us with unseen, like some examples that were not seen through the training, and we can. And what we basically want to predict y for those examples, given the trained algorithm, trained network. So we can also ask this question: is learning in general feasible? Is it feasible at all or no? In fact, it is. So we can have some mathematical guarantees for that. So imagine we can have the error for, we call it in error. So basically, in error, it's error within our training data set, with like how well our learning algorithm, trained algorithm, is performing on our training data set. And also we have error which is outside of this. That is basically the error on unseen examples, unseen access, which is here. And so actually these errors, the error within unseen examples can be upper bounded by this formula, right? So I'm not going to go into detail. The take-home message here is just the feasibility of learning. So we have this error can be bounded. Now, finally, let's move to some biology and some applications of machine learning in evolutionary biology. So here is the example how we can use machine learning to distinguish between, for example, hard sweeps and soft sweeps. We can derive from genetic data. We can derive some summary statistics, and that's going to be our input vector. And we can train algorithm on that. We can simulate some data, genetic data. We can derive those access from those simulated data sets. We can train our network, and then we can pipe our true data, true vectors, and predict whether it's hard sweep, or classify whether it's hard sweep or soft sweep. We can also do this kind of like, it's more like phylogeography / like reticulation inference problems. So there was one paper they showed that you can use some summary statistics extracted from your tree topology, and you can predict whether you have or don't have reticulation on your tree. This is one within other examples of applications of machine learning in biology, especially in population genetics. As for soft sweeps and hard sweeps, and here we can also use in deep learning. We can infer, for example, recombination rates. And finally, we came to the main problem of today's problem. That's estimation of our tree topologies. And why this problem is hard problem, and of course we all know it. So we have, like, for example, we have a set of our taxa, our species. We went to the wild and collected those species. We got genomes for those species. Then we got a bunch of problems, and we want to get their phylogenetic relationship. And of course, there are problems again. So first is like homology search. It's very problematic and very difficult procedure. We have our multiple sequence alignment estimation, which is also a hard problem. And of course, tree inference itself is a hard problem. So everything is problematic. And unfortunately, like, trees cannot be observed. They can only be inferred. So in this, what I'm going to be focusing on, we're going to try to apply deep neural network approaches to accurately infer species trees. So this is just a brief recap. So just basically like a hierarchy of different methods that are used within phylogenetic inference. So there are the distance-based, character-based methods. There is, for example, there's neighbor joining methods. This is called like algorithmic methods, or optimality criteria methods, like Bayes, parsimony, maximum likelihood, right? And this can be further classified into model-based and non-model-based. Parsimony is an example of non-model-based method. And those are probably the most like frequently used methods. Again, like Bayesian approach, maximum likelihood estimation, parsimony, and neighbor joining. And again, if you think about this, if you think about neighbor joining, actually, that's kind of like unsupervised learning approach to reconstruct species topology. So we just basically have a vector of pairwise distances and want to find hidden structure, which is in this case is going to be tree topology. Again, we can use for when we are designing new methods, we can use certain performance criteria. And one of them is identifiability. It's basically, we don't want algorithm to provide the same answer under two completely different sets of parameters. In that case, we say this method is not cannot provide identifiable results. We also have like consistency performance criteria. So basically the idea here, theta is our tree topology, n is the number of sites. So we want to see like if our number of sites will go to infinity, our probability that estimated tree topology will be equal to true topology equal to 1. And this is the example of strong statistical consistency, because like tree structure is not conventional parameter. We can only use strong statistical consistency. Of course, we can use also relative efficiency, or efficiency of a method. So the estimator is said to be efficient only if it has asymptotically smallest variance. And again, because tree topology is not conventional parameter, we can't use the like classic definition of efficient estimator. But what we can use this kind of like relative efficiency. We can compare two methods, method 1 and method 2, and we can compare which method is more efficient, which provide better accuracy, right? And then we can use robustness criteria. It's basically, we're hoping that model specification is not going to affect our inference, our accuracy. And a very practical criteria, which is scalability. We want our algorithm to speed out the answer in reasonable time, so we don't need to wait for like years and years to compute our tree, right? So those criteria, want to satisfy those criteria, of course. And finally, now we're going to be talking about the tree inference using deep learning. So motivation here is basically kind of like tree by eye. So when you look at the alignment, like can you tell like, hey, this is probably there is a tree, underlying tree that generated this alignment. So and this is, again, the alignment is going to be our X input, right? And our labels are going to be Y. And we're dealing with a quartet topologies, which means we have only 4 taxa. And for this, for 4 taxa, we have only 3 possible topologies of unrooted trees. So, or 3 labels. This is like a general pipeline. So we have basically this our quartet topology. First, what we do on this bottom branch, so we're simulating alignments along the trees, and we're training our CNN algorithm. On the top branch of the pipeline, we're trying to simulate data sets, and we want to validate our various algorithms, how they basically kind of like check their performance on that data set. So, and we use maximum parsimony, neighbor joining, maximum likelihood, Bayesian inference, and again, our trained CNN. Also, and our major criteria was accuracy. So we were comparing accuracies between those different methods. Also, we looked at the reliability measures like a bootstrap or posterior probabilities. And also we looked at the class membership posterior probability for CNN, and we'll see how we can get this reliability measure. It's actually pretty simple. So this is, I'm going to give you like a very general overview of the simulation. So first we have our three topologies, right? Three possible unrooted topologies for quartet. And so first we need to simulate branch lengths, and I'm going to talk about this in more detail in the next two slides. Second, we need to provide the substitution model, which was, in this case, we used the vector of those models, like basically very standard Gibbs-Cantor, like GTR, unrest, TIM models. And they were like, for every example, for every alignment, they were like drawn, just randomly drawn from this vector. And then we have our I parameter and G parameter within those models. And our equilibrium base frequencies were drawn from Dirichlet distribution, like so. So for the proportion of invariant sites, which is I parameter, was drawn from uniform distribution from 0 to 1. And then we had our alpha parameter that determines our J parameter. We were drawn from uniform distribution. And also we used a very simple indel model to simulate indels as well for our alignments. We used fixed rate, indel rate 0.01. And we used to determine the length of indel itself. The length was drawn from truncated zip distribution, and truncation point was at 50. So basically, we didn't have any indels that of length larger than 50.
Quick question: when you're generating these, they're ordered in some way that's determined by your simulator, right? The A, B, C, and D.
Yeah, so we provide those topologies. So we have those topologies.
Right. And I'm just curious, like, so in your CNN, is it somehow take into account that the order of the sequences shouldn't be informative?
So we're ordering our sequences at the end, always like A, B, C, D.
So the CNN does sort of get to have convolutions that are in between A and B and B and C and so on and so forth.
Not really. Like our convolutions are going to stride through every column, actually. I'm going to talk about this like in a couple slides.
Okay. I'll let you talk about it.
Yeah.
Thanks.
Yeah, that's a good clarification. Yeah. So in here we have like training data set simulation and especially branch length distribution. So we need to approach this very carefully. So, and here's why. So we want to simulate different branch lengths and want to simulate it. We don't want any overrepresentation of these cases when we have, for example, adjacent branches are very long and other branches are small or something like that. So we want more or less like equally represented trees, the branch lengths in our simulated data set. So, and assume our branch lengths are going to run from the length is going to run from 0 to 1. And the question is like, why not to use just simple uniform distribution from 0 to 1? Okay, great. So we can do that. We can, of course, draw independently the branch length from uniform distribution, and the distribution of each branch length is going to be uniform, right? So that's great. But like, unfortunately, if we look at the sum of those branches or tree lengths, if we just sum all the branch lengths together and using this uniform procedure, we are not, unfortunately, going to get the trees that are very, very short or very, very long, because the sum of uniform distribution is distributed according to so-called Irwin-Hall distribution. And as you can see here, the probability of drawing very, very, very short branch lengths or very, very large branch lengths is essentially 0. So we need some other distribution that will generate those trees with very, very small or very, very large branch lengths. So we came up with this very, very simple beta mixture distribution with equally weighted components. And here, like our uniform component here, and two other components. Basically, these two other components are drawing branches, either very, very small or very, very large branches. And here's how it looks. Kind of like ugly, looks like a saddle, weird looking distribution, which is not great because we want somewhat uniform sampling, right, of branch lengths. However, the tree length, if we plot the distribution of tree length, now we don't have, like, we have some probability of drawing very, very small trees or very, very large trees. It's not zero anymore like we had with Irving Hall distribution. So, and we want to somehow, we still, like, of course, though these distributions are not ideal, and we want to simulate our trees in somewhat uniform manner from the tree space, branch length tree space. So we basically want to simulate simultaneously uniformly from individual branches, and we want to simulate also like the tree lengths. They should be also somewhat uniformly distributed, sampled, sorry. And here, if you're curious, this is the distributions of the just two branches, sums of two branches drawn from uniform distribution. It looks like a weird, like triangle thingy. And this is very weird distribution of our beta from our beta. So basically, the sum of two branches will be distributed according to this if we use beta as a branch length distribution. And again, we want somewhat uniformly sample this thing. So, okay, what would be the approach to do that? So imagine like we just like, suppose we have our trees and we're going to spread them in some certain space, and we sample from that space. And we were thinking about like, why not to use like Holson's-Back-Hills projection, which is very, very simple, right? So we have our two branch lengths, A and C, and we have three branch lengths, B, internal branch, and D. And now we can project every tree branch length configuration on this in this space. The problem, and I'll show you why we can't use this. I'm going to show you why we can't use this projection, unfortunately. So here's an example. Assume the 0 is a branch length is equal to 0 or 1. There are three different branch length configurations here, and let's plot them within this Holson's-Back-Hills projection. So let's plot the first one. So x-axis is going to be 1 and 1. Here you go. For the second one, we're going to have 0 x-axis, and for y, we're going to have 2. Here you go. And here's for the third tree, we're going to have, again, for x, we're going to have 1, and for y, we're going to have y-axis, we're going to have 1 as well. And now we're superimposing tree 1 and 3. So, but we don't want this. We want to like somehow come up with a space that separates those cases, so that we can more or less uniformly sample from that space. So, and what we did, we came up with a very simple heuristic formulas, or like tree statistics, if you wish. So the first one is, we called it pairwise distance, that you can basically derive the branch, like cycling through branch lengths, you can derive that pairwise distance, then simple length of a tree. And then we have this, we call it like sum of lengths of neighboring branches. You can use this formula to derive that. And then you can come up with this space. So basically on x-axis, we have our pairwise distance value. And for y-axis, we just multiply length by this sum of length of neighboring branches. And we can kind of like unfold that space, this Holson's-Back-Hills space. And basically every dot within this space will correspond to certain branch length configuration. For example, this one, all of the branch lengths are equal to 1. And here, theoretically, all the branch lengths are equal to 0. we kind of like, we can sample from this space, and again, every dot will correspond to the vector of branch length, specific branch lengths. And we can use some like a grid sampler, for example, and sample more or less uniformly from this space. And of course, this approach is not optimal, but at least something better than sampling from this space. So we did that. So, and now we are simulating our training, validation, and testing data sets. So we have our training data set, we have our alignments, and we have our topologies. I'm just reminding you there are only possible 3 topologies or labels. Then there's validation data set. The validation data set we're going to use just to stop our training algorithm. So, for example, if accuracy reaching a certain point that we're satisfied with on validation data set, we can stop our training procedure. And then we have our testing. We want to predict the labels, which are 3 topologies, 1 of the 3, for this for those alignments. And what we do here, we use very simple CNN convolutional neural network approach. We have our filter, and our filter is very simple. It's the size of the filter is 4 by 1. Basically, we're striding our filter across every column in your alignment, and we perform several convolution and pooling operations. Again, we just like piping everything through fully connected network, and we are predicting the labels 1 to 3, which will correspond to our 3 topologies. And this is accuracy on the test data set, and the test data set in this case, that was a test data set derived from this branch length space. So if you go back, so from this space, right? So, and as you can see, the accuracy is kind of like very good for gapped alignments for CNN. It outperforms spectacularly other methods. On ungapped alignments, on CNN also is outperforming, although you have to provide more training data to achieve higher accuracy than other methods. I mean, accuracy is not like significantly different, but you can see that it's going up with more training examples you supply, higher accuracy you can achieve. But let's talk a little bit about the tree branch length heterogeneity. So basically, it has been known. So there are several areas of branch length parameter space when certain methods perform differently. So, for example, this is the most famous case. It's a Felsenstein zone that causes long branch attraction when we have very short internal branch and we have very long disjoint branches. So, for example, parsimony, as we all know, the parsimony will combine. We'll try to attract these branches together, and we'll infer a tree with these branches as sister. Or there's this Ferris zone. Basically, maximum likelihood doesn't perform well within this zone when we have very long branches that are sister adjacent branches. But however, maximum likelihood is consistent method, and supposedly with very large alignments, we don't have that problem. So again, one more clarification for our simulation data sets. Our length of alignment was 1,000 sites. So we also simulated different zones that can be seen here. So this is our BL space, kind of like our everything, and then we have different zones, and we use different distribution, different approaches, and we try to validate different methods within those zones. Here are some examples on gapped alignment. So, and the performance of CNN. So there's in Ferris zone, it's pretty good. So twisted Ferris zone is also pretty good. Felsenstein zone, it's not as great as, for example, Bayesian inference or maximum likelihood, of course, but you can supply more training examples from Felsenstein zone, and you can actually increase accuracy, performance accuracy of your CNN network for that examples, and you can reach accuracy comparable to maximum likelihood on alignments of size 1,000 columns. And this is another short internal branch zone. We have very long branches, and we have short internal branch. And CNN is outperforming everything else.
We're right at time now, so.
Yeah. So, and one more. This is the last slide, basically. Also, comparison of reliability measures. So we have our kind of like bootstrap support, and for CNN we have our support. So basically, if you correctly classify your tree topology, you're going to get correct. So you're not going to get small posterior membership posterior probability. If are incorrectly classified, you can get you're not going to get high support. You don't have basically positively misleading examples. So and finally, conclusions. So we have our CNN, which is very accurate, and once it's trained, we can instantaneously generate our topologies, and we can improve our CNN. The sky is only the limit. In the future directions, we can improve our architectures. We can do multi-locus inference, or we can of course try to expand, and we can infer trees on with larger taxon sampling. And thank you to all the members of the Schreider's Lab, and thank you for listening.
Thanks, Anton. So we have a question. If I have 4 new sequences of the same species, how should I determine the training data set?
4 of the same species?
I'm not sure what that means. New sequences of the same species.
It's like generating the population tree.
I'm not sure. Maybe we can, Suhao Ko can clarify. Let's see, so I guess, so my question, maybe you can just clarify in the chat and I'll ask again. But yeah, so my question was, the convolution is, I see, on the columns of the sequence alignment. And so is that sort of, I don't know, equivariant or invariant to the reshuffling of the order of the sequences? I mean, not, what am I trying to say?
Order is important, yes.
Order is important, but I mean, so people have developed neural networks that are equivariant, meaning that if you, for instance, flip A and B, then the classification flips on A and B. So is there any explicit attempt to do that sort of thing?
No, I don't think so.
I mean, is there any danger that, for instance, like if you have some consistency in the way that indelible is ordering the sequences, that could be picked up as signal in your convolution?
I haven't tried that, but that's a good question. So I was just like, our approach was very simple. Just like we're just like keeping our labels consistent across all the simulations. A, B, C, D.
Yeah, I just don't know if there's some sort of artifact, a potential for artifact there, but. So yeah, so I guess the previous question just said, yep. So I'm not sure exactly what that means. So we have a question from Steve Haddock. I think related to the question above, what are the considerations for the training set and applying it to other sequences? Yeah.
Yeah, so that is hard question. So because we don't know how nature generates our examples, right? Our sequences and branch lengths. So we did only that was like a proof of concept thing. So we only did this on simulated data set, right? So we just wanted to see that we can learn something from this weird branch length space.
So for instance, if you, what if you were in a regime where there was a different, did you try anything where you trained with one branch length distribution and then, or sorry, one indel length distribution and then tested with a different indel length?
We haven't tried that. We haven't explored that. Yeah, but
yeah, I mean, it's just or maybe sort of like simulations with selection or without selection, and then inference with selection. It's just like there's data from nature is just so weird.
Yeah, but the beauty of machine learning, you can simulate your data the way you want. You can make it as complex as you want, right?
Yeah, I bristle a little bit at that though, with just because the simulator is always going to be less complicated than the actual
of course
data, and so. I mean, there has been evidence that if you have like a really complicated inferential machine, then that can, when you have a domain shift, it's sort of less robust.
Mm-hmm.
I mean, there are situations in which it can be robust too, but it just depends. Okay. I'm also just curious about like, what are your thoughts for scaling this up to bigger trees?
Yeah. So one of the relatively straightforward approaches will be to use some amalgamation methods. For example, when we can infer quartets from your multiple quartets from the bigger tree, right? And then you can join them together into bigger tree. That's one of the approaches. Another approach that will be it's so-called online training. You can kind of with online training, you can kind of use network, and you can train it on the go. And for example, train on possible four topologies, right? So you train the network on those four topologies, you pick the best one, then you do, for example, an NNI step for that topology, right? And then you do it 4 times, and you have 4 possible other altered topologies, and then you pick the best one. And this is how you can kind of in this online learning, you can converge to hopefully true tree, or better tree. And it's scalable. It's scalable.
It is. I mean, that sounds expensive to me.
It sounds what?
Expensive, computationally expensive.
It will be expensive, but yeah, if you have access to GPUs, yeah, it's going to be pretty fast. Even on larger alignments, it's not going to take a lot of time.
Okay.
Yeah.
For more than 4 sequences, yeah. Okay, so right, so there's somebody asked the quartet question. Yeah, it'll be interesting to know. I mean, I know that some of these things are statistically consistent, but I guess to have them work properly, they have to have sort of good error properties as well.
So the problem with amalgamation methods, I know that's it's a statistically consistent method, but the problem is even if one quartet is incorrectly inferred, then your procedure can fall apart.
Mm-hmm.
That's why this I mentioned this membership. Remember the kind of a boost reliability measure for inferred trees using CNN. So it's not so basically you're not going to get high posterior probability for incorrectly inferred trees. Then you can set up a very hard threshold, and you can just filter out those quartets that even slightly incorrect.
So Andrew Roger just chatted me that a lot of problems occur for many methods when there's model misspecification. I wonder if you simulate under one model and then your test data is simulated from a different model, how robust the CNN method is, which is, I guess, similar to something I asked.
Yeah, we haven't tried that explicitly.
I guess it's also interesting, it seems like the main strength here is in the indel, is using indels properly.
Yeah, so that's kind of the idea also, because CNN is basically to construct the abstract indel model when you train your network. So, but again, the problem, all those alignments in our within our simulations and the pipeline, we assume the alignments are correct. But what alignments are going to produce as the correct indels?
But you assume the alignments are correct. Say say more about that. So you simulate, yeah. So you simulate, and then you assume that, oh, so you use the simulated model, no, the simulated alignment directly in your inference.
Yeah, so alignments are, they're true. So we use true alignments. Yeah.
So, okay, so that's skipping an inferential step that would be present in like a real data example.
Yeah, so basically, yeah, so in my opinion, it would be very hard to like our aligners are not producing very accurate like gaps like in those, right? They're not estimating in those correctly, especially like if you have very fast evolving sequences.
But okay, you're saying that it's hard to get good high quality alignment, but you're using the simulated alignment.
The simulated, yes, of course, yeah. But I don't think if it's like, okay, we simulate like everything is perfect, but when we try to align that trained model on simulated data to actual data, I'm not gonna sure if it's gonna like necessarily work perfectly. You know what I mean?
I think so. So yeah. Okay, that seems like a limitation. I guess I sort of also wonder, like, if you could compare to, I mean, even just a simple gap coded.
So in fact, like when you do alignments, they're like simply gap coded, right? So I just like replace this A, T, C, G, and gap with like numerical values. And also the when you do this filter procedure, when you like do this convolution and you stride your filter across the alignment, you can assume some dependency between you can widen your filter, and you can stride, for example, through 5 columns at a time or 6, or you can even apply entire filter for the entire alignment.
No, that's cool, and I feel like that has a lot of potential for more interesting and complex mutation models. Because like that sort of, for instance, depend on the nucleotide context.
Mm-hmm.
We should think about that, because that's something we care about. But let's see. I was just saying, for your comparison, it would, I mean, since so much is driven by the indels, and you're assuming, if I understand correctly, the correct multiple sequence alignment, it would seem like perhaps even maximum likelihood using a gap coding mod, like you can do maximum likelihood or parsimony or something with a gap character. That might be actually that might perform pretty well as well.
Yeah.
Anyway, whatever. Lots of things. I mean, there's always. I mean, you do one thing, and then everybody says, "Well, why don't you do the other thing?" So maybe we've got one more question from Roger. He says, "It seems so." This is Andrew Roger. It seems like maximum likelihood did very poorly overall, which was surprising. Worse than Bayes, for example. Did you investigate this?
No, we did not. And it's within this weird. Within this weird space that we generated, our simulated weird space. So, but we didn't investigate that further, and but it is surprising.
Okay. All right. Well, thanks for that. It was cool to get the flyby of all of machine learning. So thanks for that. That's a great way to start our series. Yeah. So looking forward to the next one.
Yeah. Have a good day.
Thank you very much.